All right, so thank you everyone for taking the time out of your day to come see whether we can make our laboratories more sustainable by reusing daily consumables. This is just primarily going to focus on the questions of pipettes and whether we should be using glass or plastic. And just to start off the presentation, I'd really like to give a shout out to Pascal Pfister because he was really instrumental to starting this off. He's one of my colleagues, a fellow PhD candidate, and really we kind of like work together on this project. So much of the work of this is not just mine, it's also his. So just to maybe, I guess, ground your expectations for this, I come from the laboratory of Professor Toby Erb, where primarily our focus is actually understanding and building metabolism. So whether this is artificial photorespiratory bypasses or the building of light-powered chloroplast mimics, our work is primarily about looking at how we can engineer new to nature reaction chemistries for the fixation of CO2, as well as this sort of building of synthetic organelles. So we are not experts in life cycle analysis and sort of like tracing all of these like complex flows of matter in the environment. We are biochemists. We have a much smaller viewpoint. But where my sort of like personal connection comes to this question is I am inspired by the use of plastic waste as a novel substrate for industrial biotechnology. So I'm working on the sort of end-directed question of once plastic becomes waste, is there ways of basically upcycling this and using it in novel ways besides just leading to incineration or a landfilling or these other sort of like attributes. So basically taking recycling and seeing if it's possible for this to be carried out either enzymatically or with microbes. But again, as I said, this is looking at the sort of end problem rather than those upstream. So I'm going to try to convince you today that we need to consider primary production. So today's overview is basically going to be looking at the plastic accumulation problem, kind of giving you a sense of the order of magnitude for those that are not kind of imbued with these statistics. I'm also going to briefly tell you about our comparison of glass versus plastic pipettes, and finally, maybe some strategies or framing for increasing the sustainability of our biological laboratories. So in case you are not aware, plastic production is actually increasing exponentially alongside our CO2 emissions. So if you look at the data from about 1950 to when a lot of plastics started to were developed and began gaining prominence to about 2015, we currently have about 350 million tons of plastic in production, and this is expected to increase 70% by 2050. Unfortunately, of this incredible mass of plastic that continues to be produced, only 9% of it is actually recycled. The remainder is either sent to incineration facilities or it's just landfilled as waste. Furthermore, this is particularly problematic in that a lot of nations with established recycling facilities have stopped accepting plastic waste from the global north. So we're kind of facing this deficit of possible plastic recycling facilities. So basically, just to send this point home, when you talk about the sort of generation of a plastic at the beginning of its lifespan to where it ends at the end, they mainly follow a linear pathway from cradle to grave. And as you can see here, these estimates that kind of root us are famous to the Geyer et al. Publication in 2017, where basically they estimated that about 8.3 megatons of plastic are currently in production. And as you can see, majority of it is discarded and incinerated. Very few is recycled. And this is due to a variety of factors that we're going to be discussing. But if you look at actually the main plastics that are going to waste and you stratify them by which industry they're from, it turns out that plastic packaging is actually the most problematic. And this is basically, or one reason that can be attributed to this is that its lifetime is so much shorter. So packaging tends to be single use and then immediately it goes to waste. Whereas if you have materials such as those that are used in building and construction, they have much longer lifetimes and therefore they stay in the cycle for much longer. So then when you look at the sort of outcomes of this single-use plastic waste and the plastic that is at the end of its life, we find that it often actually disproportionately ends up in the ocean. So it's not the mismanagement of these coastal recycling facilities, as well as just other factors that contribute to the loss of this waste can result in like hundreds of thousands of tons of plastic in surface waters, which, as we now know, is having drastic effects on the ecosystem there. So there's beginning to be studies about how the effect of microplastic fibers can influence reproduction and mortality of organisms like the Pacific mole crab. You may have been familiar with this idea that turtles are actually influenced by the plastics that occur in the ocean, and they mistake them for jellyfish, which affects their feeding and reproduction. And most drastically, in my view, is that we're beginning to see microplastics and other synthetic particles in some of the deepest marine ecosystems on Earth. So it's not only something that's affecting the surface waters, that's accumulating in, say, the Great Pacific Garbage Patch, it's something that's beginning to proliferate these aquatic ecosystems. And if the ocean is sort of not convincing to you at all, we're also finding that in very protected areas of the world, such as national parks in the United States, we're finding a phenomenon called plastic rain, where these microplastic particles are being carried on in clouds and entering spaces that human beings have touched very little, only through hiking and whatnot. And maybe most drastically is the concern for human health. We're actually starting to see the accumulation of microplastics in the human placenta. So this is not just something that's affecting the environment that is outside us, it's also something that will eventually at some point begin to affect our species. There's many discussions about to what degree this will actually have a negative effect on us, and maybe it won't, maybe it will just maybe they won't have drastic effects. Those studies need to be undertaken, but I think it's a considerable point for just how much plastics have begun to influence life on Earth. So if you look at the reasons for maybe why plastics persist in the environment for so long, we kind of amusingly see that few organisms are actually capable of assimilating plastics. The sort of select examples that people have found are the basically consumption of polystyrene by the gut bacteria of mealworms. There's a little bit of polyethylene consumption by, again, caterpillars. And in 2016, the focus of a lot of my research as well is this bacterium, Ideonella sacchaensis, which is capable of assimilating polyethylene terephthalate. So if you look at this, the reason is that a lot of plastics form these very intense repeating structures of carbon, and the bonds are not very accessible to a lot of biological enzymes such as hydrolases. So if you look at which ones are actually capable of being recycled, either by chemical methods or by biological ones, it's very few. It's basically limited to polyethylene or the type 2 or type 4 plastics, depending on whether they're high or low density, or it is this type 1 polyethylene terephthalate. So the vast majority of plastics that we are using and basically leaving this sort of circular cycle of the plastics economy end up being not recyclable. So this they tend to persist in the environments for incredibly long periods of time. So just to remind you, we're really facing this crisis where most of our plastics are either discarded or incinerated, and very few are able to be retained in this recycling loop. So the main thing that we can do to basically reduce this continuous acceleration of plastic production and consumption is perhaps focusing on the primary production. So just in general, keep in mind that plastic waste accumulates in nearly every environment on Earth, and this is largely due to our reliance on single-use plastics. And then so far, our mechanisms of mechanical recycling or organismal degradation is not really sufficient. So I would just like to pose a question of, like, what can we do as scientists involved in this? So as many of you are aware, we tend to produce considerable single-use plastic every day. And this tends to be problematic because due to a lot of the experiments that they're used for, either with very intensive chemicals or microorganism contamination, we don't typically recycle our plastic pipettes. These generally have to be autoclaved before they're sent to the landfill. So this poses a bit of an ecological problem. So if we do a sort of quick order of magnitude calculation for our lab, and you just assume offhand you're doing one protein purification experiment a week, that costs about 3 pipettes. If you multiply this by a lab of approximately 40 people, that's 120 a week. And then in the course of a year for about 40 people, you're looking at over 6,000 pipettes. So very quickly, these small pipettes accumulating in our trash bins don't seem that significant. But if you think of just a single laboratory, there's a tremendous amount of waste that's being produced. Furthermore, the real consumption is actually a lot higher than just this quick Fermi calculation. In fact, we got the numbers from our ordering system of how much our particular laboratory consumes at the Max Planck Institute of Terrestrial Microbiology, and we find that we're about 9,000 pipettes per year, and this translates to about 112 kilograms of plastic waste. So if we do a bit of an order of magnitude calculation on the amount of energy that this is taking, this additionally requires about 31 autoclave cycles if you are if you're performing and if you are exactly perfect tech and filling 300 pipettes per cycle, we measured that's as many as you can fit in at once, which means running those 31 cycles and consuming about 84 kilowatt hours of energy per day. You're consuming about 2,604 kilowatt hours in the course of this year of consumption. And none of this really accounts for the fact that there's CO2 emissions, water usage, and transport to the incinerator. So I think that these numbers kind of demonstrate that we are, as scientists, really using a lot of single-use plastic, just if you're only looking at pipettes, not petri dishes or these other aspects. So my colleague Pascal and I, we kind of wondered, okay, well, let's just compare the obvious parameters for two different pipettes. We'll use one from soda lime glass and one from polystyrene and try to see if how the price efficiency compares and also how their sustainability compares. So the main point that most scientists are going to care about is like, what are they actually accurate? There was a big debate in our lab in the beginning about whether you could trust the gradations that were with non-glass pipettes versus the plastic ones. So we did the experiment and it turns out there's no difference. And interestingly, you see that glass is actually more affordable after a few uses. So a lot of resistance comes from the fact that there's a large upfront cost to introducing glass pipettes because as you can see, if you have a plastic pipette, it works out to about 25 cents for the Falcon brand or 0.08 for Stadtzeit. But with glass, really, you have to pay a lot of money to sort of get your laboratory started with this. But after about 30 uses, there's actually a parity that's reached. So you are able to basically sustain your lab, provided that there's infrastructure for the reuse of glass. So basically, if you're looking at, if you're doing a direct comparison of this plastic versus glass pipettes, you really see a linear pathway where your plastic pipettes are essentially going to be autoclaved once they're used and then transported directly to the incinerator, and then the generation of CO2 occurs. Whereas with glass, hopefully, once the resource is already made, you can keep them in looping in a circular economy simply with the use of water and energy to power autoclaves. So one thing that you might be wondering at this point is, okay, with the energy intensiveness of autoclaving, is it actually better in terms of CO2 equivalents to switch to glass pipettes over plastic? So if you do a quick set of calculations, which we really based on a life cycle analysis from 2013, you do see that basically it is much better to use glass pipettes over plastic. So if you do a comparison, there's about 45 grams of CO2 emissions per pipette for the polystyrene version, whereas we're looking at 1.3 grams of CO2 emissions, provided that a renewable energy is used, which is the case in Marburg. So what these sort of life cycle analysis basically demonstrate is that it is actually more sustainable in the long run to use glass pipettes, but not necessarily to keep using the polystyrene ones. So I think the results are fairly clear. We can see that glass pipettes are just as accurate as the plastic ones. They're much more sustainable under the given conditions, and they're much more cost efficient over time. So when we kind of gave this presentation initially at our lab meeting, when we were debating the question of whether our lab should switch to glass or plastic, most people were fairly convinced. They said, yes, we can definitely do our experiments with glass pipettes, maybe have a few plastic ones on hand for things that are particularly sensitive. But there was still a bit of a resistant group that said either, no, I don't think we should switch, or I don't think we should switch at all. So we kind of wanted to probe a little bit more about why that resistance existed. So basically the concerns that emerged from our survey was that scientists are really concerned with the sterility of the glass ones, considering that they're all in a bin or in a tube autoclave together, the time associated with cleaning, and basically having the available infrastructure to ensure that there's a continuous turnover of supply, rather than just being able to go to the wall at will and pull out a plastic one. So amusingly enough, I think that even if you develop the infrastructure, say by communicating with your autoclave facilities, and basically ensuring that there is a system for sufficient turnover, and ensuring that they're sterile, and all of these other aspects about the fact that there's less CO2 equivalents that are produced. These facts alone are not really enough to transition from a sort of efficiency-based competitive mindset to a systems mindset. So one of the quotes that I wanted to draw your attention to from our survey is a scientist expressing that they needed to be fast, and they just didn't want to waste time sort of dealing with the infrastructure associated with using glass pipettes. pipettes. They write that it was simply not practical to propose a system, and this is a reason why plastic ones are invented in the first place. So obviously, I think this quote reflects a deep concern that a lot of scientists have, that incorporating these systems that maybe are more sustainable in an abstract sense has the tendency to slow down their scientific research and their ability to be competitive on the global stage. So I think that if we really want to go towards I guess implementing the strategies that we know to be better for the environment and we know to be better for the climate crisis in the long run, we have to address these sort of ideological barriers to adoption. The one thing that I would suggest in general is we need to reconceptualize the scientific lab as a part of a larger ecosystem, rather than something that just is there to generate data and kind of exists as its own isolated environment working on problems. So with that, I would just like to thank you for your attention on this particular problem. And yeah, again, thank you so much to Pascal for like pioneering this in our lab, and also Toby for giving us kind of a platform to explore these sustainability questions and the concern in them. And of course, all of my colleagues. So yeah, thank you for your attention.
Okay, thank you very much, Victoria, for this great talk and the insights and what you figured out about glass and plastic pipettes, and to see why it might absolutely make sense to consider switching from from plastic to glass pipettes, or at least in particular, right? And I think another aspect, I just discussed it yesterday with a former colleague of mine, is sometimes also the availability, because especially now during Corona times, they ran into shortage and they couldn't get delivered the plastics pipettes anymore. So they were happy that they had some glass pipettes also on hand, which they could just reuse and were not completely dependent on the plastic one support. So thank you very, very much. There's already a lot of people writing in the chat. Thank you so much for the talk.
Maybe I'll stop sharing my screen now as well.
Yeah, you can do. Mm-hmm. So whoever wants to raise a question, there's basically two pretty good options. You can either raise your hand, as Victoria also did now. And we'll happily unmute you and you can ask your question directly. Or there's this question and answer bubble on the bottom of Zoom where all the questions can be collected, because sometimes when a lot of people are writing into the chat, then questions can get lost in a lot of other comments and so on. Yeah, there was a raised hand, now it disappeared, so don't know. I'll give you a small moment. There's a question on in the question and answer bubble already. What about pipette tips? What do you think?
So this is something that we didn't really address. I know that there's some laboratories that are working on ways of recycling pipette tips. I would state that as a scientist, you would probably need a very extensive sorting system for that afterwards, just to reduce the contamination of using a particular chemical that could be toxic or could really affect an assay. I think for me personally, as a biochemist, I would have a really hard time reusing tips just because of that uncertainty of ensuring that they're completely clean. But I know that there's people that are working on this. So I can't comment on that because that's not something that we looked at in our lab. We just thought that glass pipettes are the easiest to use, because it was it's something that a lot of students are taught. Actually, I know in my bachelor degree, for example, all lab work was done with glass pipettes, and then you sort of emerge into your professional scientific career, and you have these plastic ones on hand for this sort of ease of use and efficiency. So basically, I guess we're trying to argue that it's very possible to use these glass pipettes that are readily available. It's just it's kind of a training matter, and also having infrastructure at your particular institute for them.
Have you also used, I saw that there's also sometimes glass pipette tips available. Have you ever used these ones?
I have never used those. I mean, I think you would have to be remarkably organized just so you wouldn't break them. I think it's definitely possible. It depends. Yeah. Especially in labs that don't go through hundreds of pipette tips a day, or somewhere where you're doing, say, massive enzyme screens that require hundreds of pipette tips. I think it would make a lot of sense to employ them in those particular instances, maybe rather than the just day-to-day.
Then also Nicoline, she commented on this question. There are two commercial systems for pipette tips as far as she knows. So from Granova and Ion Field System. And what I know, also some of the pipette tip distributors, they use at least now recycled racks and so on, and some of them are also collecting them. So sometimes it's interesting to see on which kind of suppliers you're using, and if they already have maybe a recycle system at least on hand for pipette tips. Okay, then the next question in the chat: any suggestions on how the same analysis could be done in our labs?
I would just suggest maybe like, again, getting the initial numbers on production from whoever's responsible for ordering. And then in terms of like I don't think the accuracy tests are really necessary to complete because they're reported by the manufacturer and generally quite trustworthy. Maybe it's important for sterility. Like I see there's a following question about someone asking if we tested sterility in cell culture. We don't have cell culture in our laboratories just because we primarily work with bacteria and yeast. So I think like that's something that would be very important, especially because cells can be so sensitive, and like you always have to worry about like mycoplasma contamination, all these other aspects. So I would guess that you would need a much more intensive detergent maybe, or a longer sort of washing phase afterwards, just to ensure the sterility before you put them into the dishwasher and the autoclave. Yeah, that's I think you have to kind of make it specific to your lab and see like where it's possible to maybe use these things and where it's not. Like I know maybe in the case of really sensitive like S2 organisms, it just might not make sense to like economically to like do all of this additional work. But I think definitely for routine experiments with bacteria, it's totally possible to employ this sort of system.
And maybe also the other questions following up. So how do you actually clean them and do it in the autoclave, and how you store your glass pipettes?
Yeah, so obviously they're stored in their own sort of like metal canisters, which can then be autoclaved. But the sort of initial steps after usage is we have like this large plastic bin in every single lab that's filled with this detergent solution. So pretty much immediately after pipetting LB or a bacterial culture or whatever, you place them in there. And this acts as a sort of like pre-sterilization step that just generally prevents any sort of media from sticking to the pipette and sort of temporarily neutralizes any sort of bacteria that's on there. And then we basically put them in the dishwasher and then the autoclave, and then they're ready for use again once they're packed into these metal containers.
Then there's a question from Jakob. Did you now switch completely to glass pipettes in your own experiments?
So I have switched completely to glass for my experiments because I think it would be quite hypocritical if not. But I will admit there are cases when I've been doing a particularly challenging genome knockout in Pseudomonas and like struggling a little bit to get the experimental results that I want just because of contamination. So sometimes I use plastic in very few cases, but like for all E. coli work, it's glass.
So did you check if there could be microplasm in the glass pipettes left over from a contaminated cell culture after washing and autoclaving?
I think that's a really important test for anybody that wants to use them in cell culture. We didn't, just because we don't employ cell culture in our experiments, but I think that would be definitely a useful test.
And what is your opinion on autoclaving glass at institutions that don't use 100% reusable energy? Energy mix in Germany is about 350 grams of CO2 equivalents per kilowatt hour, and our medium autoclave uses like 5 kilowatts per hour. At the moment, we use glass pipettes. Now it jumped, sorry, only for non-sterile applications for that reason.
Yes. My opinion. I think like you can definitely go through and sort of make these rigorous like energy calculations, but I think that's like a sufficient deterrent for many people about these systems, like these circular systems of reuse. And I think that that's a fair argument. Where I kind of fall down on this is like, as someone that's trying to like engineer bacteria for the degradation of plastic waste, you see just how challenging that is as a problem, and how currently just we don't actually have good solutions for any of this. Like I said in the beginning, like we're just we're sending the waste off to an incinerator or a landfill and then just choosing not to think about it. But I think like with the rate at which like CO2 emissions are accumulating, like we're starting to not have options of just not thinking about it and constantly treating these like production pipelines as linear. So although I think that it's a very decent counter argument to say that like, okay, the energy mix is not perfect, so why would you additionally run the autoclave? I also think that like it's a bit of a luxury to not think about where all of our plastic waste is going. And again, this is only pipettes. This is not considering falcon tubes. This is not considering so many other aspects of our research that really produces a tremendous amount of waste. So I just think that like, if there is something that is like as low barrier as possible to change, then I think that in terms of sustainability, we should start looking at these things so we can move towards this goal of making our institutes carbon neutral. So I guess like I come down on that in the sense that like the larger perspective is we don't have the technology currently to effectively deal with this problem of waste. So I would always favor the reuse, even if it's like, even if the energy that goes into that is like slightly more, because I think that the sort of large problem in the back of our minds is just that we have the luxury of suppressing this and not thinking about the long-term consequences of this. I mean, that's much more political as an opinion. So I will fully express that. That is not entirely a fact-based argument, but that's where I stand on that.
I mean, maybe also an argument, especially when you consider costs, is what Nicolene brought earlier in the chat, is that you also need to consider the salary of your stuff, right? Which needs to autoclave the stuff, which needs to, maybe also goes in hand with the question, which is still in the chat of cotton plunging of your pipettes and so on, right? So also this might increase the price depending on where you are and how the salaries are. But I guess, as you say, right, sometimes it's also worth paying a little extra, and then it's not as cost efficient as it might be, but at least you don't have the waste piling up, right?
And that's sort of like the larger question that I want to ask in this presentation too. It's not strictly about glass versus plastic pipettes. It's about this question of like, what role do scientists have? Because like, I find it tremendously ironic in my own research on plastic degradation that like you end up producing so much waste in the course of a PhD. And like, yes, you're working towards these ultimate goals, but in the intermediate time, it can be like very difficult to address those problems. So I guess like it's more of a call to action that like we should do as much as possible like in our institutes in the little ways that we can, just to move towards this goal of a more sustainable future.
I think also an interesting question which just popped up in the question: did you have any general problems with broken glass? I mean, in case of security and so on, right? Because that's also something I was wondering. I guess it's also an interesting question. For sure.
I was going to say in the beginning, yeah, we definitely, this was something that we had to address like one lab meeting after the sort of introduction, is just like, be careful. So I think like there's kind of a learning curve with any new adoption of technology. So yes, things get broken, things get damaged. Luckily, no one like pierced their hand or did anything terrible, but yes, I think that's fair. Maybe also, Jan, in terms of the like CO2 analyses, it would be worth to share that study from Bioarchives that you also shared with me. Somebody recently did a life cycle analysis, and they also made the argument with that when you consider tech salaries and like the energy utilization of autoclaves and these things, they basically analyze the problem in a much more thorough way, which is currently on Bioarchives and very cool. So maybe if people are further interested in this, it would be worth reading their paper.
I guess you're referring to the one from Martin Farley and
exactly.
Nicolene just posted it also in the chat earlier, so it's definitely there. Benoit Nicolet, I hope I didn't slaughter his name. So Nicolene posted it also in the chat, the one you're referring to. So whoever's interested in this study, I think it was just released a couple of weeks ago, go and check it out on Bioarchive. It's definitely worth having a look at. Yeah, also Nicolene posted another one for a microbiology paper where they also analyzed the plastic and showed how you can minimize your experiments and therefore use less plastic because of minimalization and what else you can do in order to use less. Yeah, there's also one paper that Kerstin just posted about the reuse pipette tips with the Greenova system.
And they can
save about 50%. That also sounds great. So I think there's a lot of stuff coming up, right? You can see for the suppliers if they have some recycle system on hand. You can check on, yeah, if you have the possibility to minimize your experiment, right? I mean, the best plastic or the best lab equipment you use is the one you don't use probably. So if you run in a 96 well plate instead of 96 petri dishes, then you also save a lot of plastic, I would say. And I mean, that's also in one of the papers, I think it's the one from the microbiology lab. You can see that, for example, for a colony forming assay, you can get the same quality of results by just minimizing your experiment. Okay, are there any further questions? I'm also happy to, if anyone wants to raise their hand, they can freely speak after I unmute them. Okay, Jakob is also making some commercials for our wiki, where we'll also post the documents and the links which were just in the chat. So everyone can join this wiki from the Max Planck Sustainability Network. You just need to register, and you can find there a big database of all kinds of questions answered for going sustainable, either as a Max Planck Institute, but also outside of Max Planck. You can find a lot of good tips, tricks, and whatever you can use in your lab. Also, just one more reminder, feel free to sign up on our website for the newsletter, as well as joining the network. We're happy for everyone. I think it's a great community and it's great exchange going on and a lot of great projects. We have now more than 40 sustainability groups all over the Max Planck Society. And we are happy to have a number growing and soon covering all Max Planck Institutes over Germany. Yes, there's another question. Do you think this awareness of sustainability is possible to integrate in other laboratories in other countries?
I sure hope so. I guess that comes down on that point that I made earlier about this sort of efficiency mindset, rather than viewing the laboratory as this like holistic part of the entire economy and human existence. I think that, yeah, definitely there will be cultural values that maybe conflict with the adoption of sustainable ideas, but I think that's also the importance of sharing these where you can. Yeah, that's not a very satisfying answer. I wish I had more knowledge, I guess, about which countries it would be particularly pragmatic and the individual barriers that are facing against that. But I guess in Germany so far, it seems people are very open to sustainable options. And yeah, and also I apologize in advance if I butchered the German on any of those slides. I was just trying, yeah, I had colleagues look them over, but I'm sure I made a few mistakes.
Yeah, also we have on our website, there is a list of worldwide initiatives, which you can check out on our website and get in contact with them. And if yours is missing, please let us know. We're happy to add them as well, and to grow the network and to get an exchange and yeah, all that. Maybe also, because I missed a part of Jen's question in the chat, do you maybe have also data on, for example, glass bottles or any other besides pipettes, or you for now focused just on the glass pipettes?
We just focused on the glass pipettes because, again, that was like the lowest threshold to making a change. But I think like recently we've been talking about whether we could do glass petri dishes. And the question is just maybe how that would maybe work, whether we would have to be so much more careful, if breakage would be an issue. So like we're kind of just like looking at that now. It hasn't really, it hasn't taken off yet. We don't have any data to support that at the moment, but there's many different alternatives. And I would like to just like reiterate the point that Jan made about like the best way to reduce, the best ways to reduce is like the experiment that you don't do. Like I think, not that we should do less experiments, but just that like, I think it should be an aspect of like how we carry out our scientific lives, I think, is also thinking about like, okay, is this experiment really need like 27 dishes? And especially with the rise of like high throughput biology, right? It's like this tendency to like continuously think bigger and bigger and bigger, but maybe there's a way that statistically you can get the experimental results that you want without also leading to this tremendous accumulation of waste. So I think that like, it's just considering the fact that all this waste has to go somewhere, and we do play a role in this.
Absolutely agree. There's another question from Jakob. It's maybe a question of the time also. Do you have any specific recommendation for single-use corona tests, recycling, or maybe another design?
I saw someone that made them into earrings, which I thought was hilarious. Um,
Uh,
Single-use
Positive
corona
ones and
test.
negative ones.
Yeah, I think it was like a jokingly like a positive test, but I
Oh,
mean,
I
I
see.
really hope they weren't walking around with displaying their positive tests. Recycling or another, or maybe another design. Um, you know what? The funny thing about this is like I look at these corona tests and I'm like, this is like the perfect opportunity instead of doing all of these like massive uh instead of doing all this grand incineration, like why is somebody not studying like the microbes that we could like? I think the perfect view for me is like if we could have a bioreactor in the future and just like throw in waste, and there would be this beautiful consortia of bacteria that like we're able to feed off of the different types of plastics, or maybe have systems for inactivating viruses or other biological materials that were problematic. So I don't have a single recommendation for single-use COVID tests at the moment. I wish that an aspect of design and material making was considering the end result, because a lot of time the way recycling pipelines are complicated is the inability to separate different materials from one another. So they just all end up getting thrown either in the trash or getting incinerated, because it's just impossible at the recycling facility to make those separations. Like a perfect example is actually peanut butter jars. Like everybody thinks that they're recyclable, but they're actually lined with a nylon that is non-recyclable. So even if you do all the effort of like eliminating the contamination, which is already another issue for recycling pipelines, you still can't recycle peanut butter jars. Which, I mean, this is also coming from the North American perspective, because I think all of our peanut butter comes in plastic, whereas here I see a lot of glass. So maybe maybe disregard that point. But yeah, I think most of it comes from like these things should have been thought of at the beginning. But I see like with COVID, obviously the desire to get these diagnostics out and to get control the population kind of takes precedence. So I guess this is also another reflection of kind of like disaster capitalism in a way, and the fact that like when you have to keep on top of these on top of these issues that are facing people and causing like terrible amounts of damage, like these considerations of sustainability always get pushed to the back burner. But I would like to see a world in which those are also a part of the conversation from the beginning.
Yeah, I think regarding the peanut butter glasses, I just read an interesting article that, I mean, if you compare single-use plastic containers to single-use glass containers, the energy consumption of the glass is even worse, right? Because to melt it down again and to make new glass and so on is so energy heavy. Of course, you don't have the plastic, which might end up somewhere else, and you have all the microplastic, but also something to consider, right? So if you have really good recycling system that the glass is just cleaned and can be reused, let's say have a beer bottle, right? This is even better than considering the single-use plastic versus a single-use glass. There was
Reuse
another
is
thing.
preferential over recycling.
Exactly, exactly. Do you have any recommendations for cell culture activities? Plastic lost to clinical waste. Could waste hauliers do better?
Oh my goodness. That's such a loaded question. I wish I had recommendations for this. Honestly, I don't really have an answer for you, because especially with clinical waste, I really understand the perspective of hospitals and diagnostic facilities that you do not want to recycle your, I don't know, syringes and spill some virus or everywhere. I really understand the necessity of autoclaving facilities, but like to me that's why it also underscores that when we're doing like scientific research on samples that really are not that don't pose a threat to like human health or the environment. Like if you're doing some protein purification experiment in E. coli, like realistically, the chance you would probably have a plasmid in there that has an antibiotic resistance cassette anyway. So like really the chance of you needing to use a plastic pipette to carry out this research versus glass is like so small. So I think it's like also putting things into perspective. Surely there's certain aspects of our society where we can't really we can't gamble with the single-use plastic in diagnostic facilities or hospitals or things like that. But on this topic of like, could waste haulers do better? I think like it's also that we have underdeveloped recycling facilities in a lot of the Western world, like many like emergent nations like that were like, for example, I'm thinking of not necessarily an emergent nation, but in China, they stopped accepting plastic from abroad in 2018. And this like really has resulted in accumulation of a lot of plastic in many different nations because they were relying on the consistent buying of that plastic from these recycling facilities abroad. And then as soon as that's banned, like now we suddenly have this excess of plastic that just has to be landfilled. So I think like the infrastructure that like every single nation needs to develop to deal with their own waste is also significant. So could waste haulers do better? I think in general, we shouldn't be like wasting like gasoline and all of these petrochemicals in the transport of plastic anyway. I think that's kind of absurd. So I think like we need to also pressure our like governments and our own facilities to see if there's like ways of reducing the transport of waste elsewhere and dealing with it in-house in our own facilities. I suppose that's kind of like unsatisfying, but I guess like I think it would be cool actually if all science labs had like their own waste management like sub facility and they could deal with kind of things like on site, because I think that that's it also like reinforces this like out of sight out of mind. Like you don't need to worry about your waste if it gets trucked off to somewhere that you have no conception of, no sense of the order of magnitude.
Yeah, I think also an important point is right that we see that we increase our recycling where we cannot reuse even better, especially for plastic. And also not just exporting our plastic then to re-import the repetware or whatever, right? So
yeah, and particularly with
point.
polyethylene terephthalate, like it can't compete with virgin plastics on the market. Like the market value of recycled plastic is actually lower, and that's why a lot of people that are in the like biological recycling field are so curious about these upcycling questions of like, can you turn it into a bioplastic, like a monomer like beta-ketoadipate? Can you turn it into a value-added chemical? Because the products that are coming from plastic, just recycling those monomers and then stitching them back together is not economically viable. So it really, there's so many barriers even just to recycle plastic and to make that economically viable. So so really if you can stop at the point of production, it will hopefully reduce the issue a lot.
There just popped up another question in the chat. Do you know if politicians are already doing something for sustainability in research? Regulations, prohibitions, supports?
Well, I tend to follow Canadian politics because that's where my heart is at. But I do know recently, say like the politicians of France banned like single-use plastic wrap for fruits and vegetables and like grocery store stuff. So I think like that would also be cool to see. I don't know of any regulations that are specifically governing research. I think in general we tend to. I think Jakob has a point that he would like to add.
Yeah, so actually, this was an issue which was kind of neglected in the past. Politics actually considered sustainability an issue in public institutions, but not in publicly funded research. So we were kind of out of sight, and only since recently this became an issue. And for example, the Green Party is pushing this agenda quite a lot. And since we have a new government, we are actually awaiting in near future new legal regulations which will make sustainability measures in research and science easier, but it's still a big issue. I'm actually just answering a question, an email by Clara Tepol from Florida, which she was already waiting for some time because she had a very similar question. And for example, spending more money for sustainability measures, for example, recycling paper instead of conventional paper is still an issue. And together with the general administration, we try to kind of push limits and boundaries. A very interesting example is actually indeed France. So I don't know the details, but what I learned from colleagues from our sister initiative, Labos 1.5, is that the political and legal regulations in France are much more advanced for sustainability in science than here in Germany, but I can't tell any details, or I don't know any specific details. So things are changing, but in my opinion, not fast enough.
We're pushing for that, right? Good, thank you very much. Yeah, any further comments, questions, anything? Wait another moment. I think it's a really interesting topic and really highly discussed topic. And I mean, also seeing the degradable plastic, right? But if it ends up in your bio waste, it can also not be really, they don't let it degrade because they're not sure if it's degradable plastic or not. And then they just better put it in landfill and all that kind of stuff. You were mentioning the bacteria that might digest plastic at some point in time. Do you, how likely you think it is that we can see something like this coming up, or how soon?
Ooh, I mean, there was a very recent publication coming out of the United States Bottle Initiative in this past summer, August, that actually demonstrated a Pseudomonas strain was capable of taking polyethylene terephthalate, degrading it down to its monomers, and then upcycling that to a nylon. But this all didn't happen within the membrane of the bacterial cell. They kind of separated it between chemical processing and biological processing, where first they had a pre-treatment step of the plastic, then they had BHET, which is kind of this a slightly longer chain composed of terephthalic acid and ethylene glycol, and then that was then degraded by the bacteria, and then it could be converted into beta ketoadipate, which then underwent further chemical processing to this nylon, this bioplastic. So I think there's really cool proofs of concept and demonstration. It would be exciting to see those being scaled up. I also know another major competitor in the field is Carbios, coming out of France, where they're basically doing enzymatic degradation of plastic. So they have these insane bioreactors of many different enzymes, and they actually published last year a really exciting one about this LCC enzyme and increasing its thermostability, and the strategy by which they achieved this was kind of novel. So I think that there's lots of people that are interested in this concept. You could even see there's a spin-off of Ginkgo Bioworks, Alania, that's working on the degradation of plastic waste. So I think this is beginning to be kind of an exciting topic, especially within synthetic biology spaces. So I definitely think these things are coming, but there's always a difference between the sort of waste degradation that you can observe in a flask in a lab versus a 10-liter bioreactor versus your next 250-liter scaled up. So for these things to be commercially viable in the long term, there's still so much work that needs to be done. But I think, yeah, things are moving very fast, and I think the field is very exciting at the moment.
Cool. Thank you very, very much. Okay, there were no further comments or questions now coming up. So with this, I would say thank you so much for your time, sharing your data, sharing your knowledge. It was a great pleasure to have you here.